Hey guys, and welcome to our Micro Lab Exam 2 review video. So in this video, I'm going to be using the study guide that is posted on Canvas. I'm going to be using it as a backbone, kind of just like all the other review videos, and I'm going to point out the highlights to which labs you should focus on. So the labs that you should focus on are definitely the gram-positive and gram-negative lab. Those are probably the biggest ones. Also, the disinfectants one, I went over what you should have received or gotten from that lab, but I'll also touch base about it in here. And then also the idea of the unknown, like how to use a dichotomous key or how to use a flowchart. So on your test, you are going to be given some cultures. So like you might get an image of this plate, and then based off of that, you would need to tell me what species of bacteria have we seen in lab, only the bacteria that we've seen in lab. So I think there's like 12 that we've focused on. So of those ones, what species or what genus typically exhibits a beta hemolysis? And then throughout the test, you'll have about, I think it's like 6 or 7 cultures to identify and to interpret. And then from that, you're going to use a dichotomous key or a flowchart in order to tell me which bacteria do you have based on you interpreting the test results. So kind of like what you did for your unknown. So like you got cultures and you interpreted the results, and then from that you did process of elimination. But I will already give you the flowchart, and based on the flowchart you would need to select what species of bacteria is it. So I know it sounds intimidating, but don't worry. If you know majority of of the gram positive and gram negative lab, you're going to do very well on this test because that is where majority of the concepts are going to come from. So let's go ahead and start with the gram positive lab. So the gram positive lab, we talked about hemolysis. So I would expect you to be able to identify these plates in terms of what type of hemolytic are they? Are they beta, alpha, or gamma? So beta is complete clearing. So this is where like you'll see no redness present. Alpha is where there is some clearing, so you don't see a lot of red. You see more like brownish color. So that's partial clearing. And then gamma is where there's no clearing at all, but there's still growth present. But as you see, the plate is still red as compared to the beta, which is where the plate has lost its red color. So that's in terms of hemolysis. Now for the PAA, you need to know what does it select for. So all this information can be found in our protocol. So let's go ahead and pull up our protocol. So PEAs, just kidding. Sorry guys. Where is it? I don't see it. It's not in 7. Well, anyway, it's selected for gram positives, as we found out in our what's it called? In our unknowns, we found out that PEAs is selective for unknowns. So actually, let me pull up the unknown protocol real quick so we can go through that as well. Oh, and then also I'll ask no questions about parasites. I can't stand parasites. Okay, so where does it say it? Okay, so PEA auger right here. So remember that MAC and EMB, they're selective for gram negatives, whereas PEA, they are selective for gram positive. And what they do, the reason why they're selective for gram positive is because they have alcohol in them, and that alcohol will inhibit growth of gram negative bacterium by preventing their DNA synthesis. So definitely keep that in mind in terms of PEA. So that is the answer for the second question on the review. So PEA is selective for gram positives. And then the next question is going to be how interpret mannitol salt agar. So just a heads up, guys, you're gonna need to know all the media, like what is the purpose of the media, and then also what is the mechanism of which the media is used. So the mechanism for the PEA was the fact that it had alcohol present in it, and with that alcohol present, it inhibited gram-negative growth. So with mannitol, so with MSA, so that was also in the gram-positive lab. So the gram-positive lab, so MSA, the in the pH indicator, it's pink. So whenever their color change to yellow, so the plate itself is normally a pinky red, but then if the media does change color to yellow, then that means acid is produced through fermentation. And what is being fermented is the carbohydrate mannitol. So MSA is a selective and differential media. So it's used to isolate organisms that can tolerate high salt. So if the organism can tolerate high salts, then it's a hemophil or a halophils, and then that is what it's selective for, but it differentiates because it allows organisms that can ferment mannitol. So that is how it's differential and selective. It's selective for halophils, and then it's differential for mannitol fermenters. Okay, so if your bacteria cannot grow on a mannitol plate, then that means that it cannot tolerate high salt. And then if you don't see that color change, then it doesn't ferment mannitol. So definitely know that. And then also know what bacteria did we see in lab that grows well on a mannitol salt plate that produces a positive fermenter. So if it's positive for fermentation, it should be yellow. And what we saw in class in terms of MSA was that Staph aureus was the only one that was positive for mannitol. So I'm going to also ask you, based on results of a plate, what is the bacteria that we saw in lab that actually presented that result? So for example, so if I were to show a plate, so let me get my pen so I can color. So if I were to, let me open up notes for y'all. Just disregard my notebook. So if I were to, so let's say that this is a picture, and in the picture, the plate was yellow, and it was originally an MSA plate. And then you can clearly see some growth on there, so like we have some colonies on here too. So if I were to show you an image of that, I would want you to tell me that it is mannitol positive, that it's mannitol positive, and it's growth positive. So if it's growth positive, then that means that that bacteria, so this bacteria, can be classified as a halophil. Okay? So from the ideal results, guys, the ideal results, so not what you actually saw on your cultures, but from the preferred results, what would have happened based off of those results, it would have been Staph aureus. So if you saw a picture with a yellow MSA plate with growth on it, then it is a Staph aureus. Okay? So that's what I want you to do with all of these. So I want you to know the ideal results. So don't go off of your stuff, because that's not what I'm going to be testing you on. I'm going to be testing you on what should have happened. Okay. So with that, let's go back to our review. So now we're going to talk about the catalase test. So the catalase test, what plate do we use for that? So that's what you need to know. So for the catalase test, we use the TSA. And then what you do for catalase test, you add hydrogen peroxide. And then with that, if you were to do that, add hydrogen peroxide, and it was positive for catalase, you would see bubbles. So let me pull up an image of that. Oh, I don't know why my computer's being so slow. So you would literally see bubbles. Such as this on your plate. So if it is catalase positive, you will see bubbles. If it's catalase negative, you will not see bubbles on your plate. And in order to test for that, what you'll do is that you put hydrogen peroxide on top of your bacteria colonies. And then from that, you can state which one it is. So the two organisms that we looked at in lab were Staph aureus and S. pyogenes. So Staph aureus is positive for catalase, and then Strep pyogenes is negative. So if I were to give you a picture, such as what I just showed you, if I was to give you a picture like this and be like, this a catalase test was done, what is the possible organism that will produce a positive result? Then from that you would state Staph aureus. Okay? Okie dokie. So that was for the catalase. Now let's talk about the starch hydrolysis. So starch hydrolysis, that is this image right here. So on this plate, so this is a starch plate that we did, and then you streaked it, and then what you add is some iodine. So you will add some grams iodine to it. You let it sit for a while, so like 5 or 10 minutes, and then after that you look at it to see if there is a reaction. So this halo that you see right here, that halo means that it's positive for starch hydrolysis, whereas this one is negative for starch hydrolysis. And the bacteria that we saw in lab that produced that was B. cereus is positive and Staph aureus is negative for starch hydrolysis. Okie dokie. Next, we are going to talk about EMB. So EMB is this plate right here. So EMB is a differential media. What it does is that it allows for gram negatives to grow on the media. And then also, if a bacteria that is growing on the EMB plate, if it is a strong lactose fermenter, it will produce a shimmery green color, and E. coli is the example of that. So E. coli will produce a shimmery green on an EMB plate. Okay? Yes, so E. coli will produce a shimmery green, and then it's fermenting lactose. So it has lactose fermentation. Okay, so that's all I'm going to say for EMB. For Max, I'm going to write it in over here. So Mac plates. Whoa, I don't know what happened right there, guys. It freaked out on me. Okay, so for Mac plates, so let's go and look to see what Mac plates do. Oop, wrong. Wrong protocol. Okay, so Mac plates, they're also selective for gram-negative bacteria. So they will grow gram-negatives only. So let's write that down. So they grow gram-negatives. And what else do they do? Let's see, guys. So with Mac plates, if lactose is being fermented, there is an acidic condition on the plate, and then with lactose being fermented, there's a color change also within the plate, and it turns very red, and the colonies will become positive. So they will become like this fuchsia looking color, like fuchsia, pinky red. It's kind of an ugly color in my opinion. It kind of looks like bloody snot. So if these colonies turn colors, if they turn red, then they are positive for lactose fermentation. So with that, red colonies equal lactose fermenters. Okay. And then what else do you need to know from this? I believe that's all you need to know in terms of MAC. But also, I guess also know the pH indicators. So the pH indicators for EMB and for MAC. So for EMB, the pH indicator is eosin, sorry guys, can't pronounce it, eosin methylene blue. So that is the abbreviation, but the two indicators in it is eosin, so that eosin right there, and then methylene blue. Those are the two indicators or pH indicators for EMB, and then for MAC, it is neutral red is the pH indicator. So definitely keep those in mind as well. So you need to know the components of each media, and those are the components that you need to know for those two at least. Okie dokie. So let's go ahead and move on to our next media type. So we're going to talk about TSIs. So there's a lot of stuff with TSIs. So TSIs is in regard to this image right here. So that image right there, definitely be able to, out of all, I believe it's 7, out of all 7 of these tubes, you need to be able to tell me what's going on in each of them. So one has a red slant and a black butt, one has a red slant and a yellow butt, some have some bubble production, and some are all yellow, some are all red. You need to tell me what that means. And what that means for each of them is right here. So definitely know this, guys. That's very, very important. So on the test, I can pick any of those 7 and put them on the test. So you need to know all of them. You need to know what having a yellow slant means, or having a red slant means, a yellow butt, a orangey yellow butt, gas production, and then also blackening. What does that mean? So definitely be able to tell me that. And then also the pH indicator for TSI is phenol red. So phenol red, also know that too, guys, okay? So if you need to, pause the video so you can read over all of that, but I'm going to go ahead and go on to the next media. So the next media is going to be the SIMS. So the SIM test tests for 3 different things. It tests for motility, indole production, and as well as, I believe, an enzyme presence, if I remember correctly. So with SIMS, you need to add Covex to it in order to test for indole. So let's go ahead and go to wrong one. Let's go to the SIM. So with the SIM, you're looking for, like I said, you're looking for H2S production, motility, and then indole production. In order to test for indole production, you have to add a Covex reagent. And then if indole is positive, you will see a red ring at the top of your tube, and that means indole positive. But if no color occurs, then that is indole negative. And then if you have a black tube, such as this one right here, if you have a black tube like that, that means that there was H2S production was occurring. And then the enzyme that is being tested for is tryptophanase. I believe that's how you say it. So that's this guy right here. So tryptophanase. So that's the enzyme that you are looking for whenever you are using a SIM test. And then also for motility, you're also looking at motility. So if you were looking at motility, then you would have your test tube, and then you would have your media. And then if there was motility, then what would happen is that you would see bacteria like kind of scratchy throughout. So that means that it's motile positive. So that's positive for motility, Whereas if it was modal negative, then what would happen is that you would only see like a line where you put your needle. So that's modal negative. Okay, guys, so that has to do with SIMS. The bacteria that we saw for that, we saw quite a bit actually. So let's go ahead and just pull up that. Oh, I don't have it pulled up. Let me pull it up real quick. So sorry, guys. I believe that was number 7. Here we go. So for SIMS, you can look at that row, and we looked at that for all different types of bacteria. So that's all fair game, but I'm going to tell you right now, I just want you to be able to interpret different SIMS. So if I were to give you a SIM that was a black tube, I would want you to tell me that that it's positive for H2S production. Or if I showed you a tube that had a red ring at the top, that means that it's indole positive. And then also the Covex reagent, what enzyme is it testing for? And it's looking for that tryptophanase. Okay, so with that, let's go ahead and go into PAA. So PAA was a slant that you had to add ferric chloride to, and if you added ferric chloride to it, sometimes you would see a color change occur. And let's see what that color change means. So I believe that was also on this one. Yep. So PAA, what this does is that it tests for the presence of phenylalanine deaminase. So that's a really, really hard enzyme, but that's what it's testing for. And then this test specifically helps you identify Proteus, Proteus bacteria. And like I said, whenever you add the ferric chloride, it's looking for that enzyme. And then if that enzyme is detected, your tube will become green in color. And then if not, then there will be no color change, and that means that it's negative to have this enzyme. So I definitely want you to know that. And I believe that's as far as I'm going to go in terms of PAA. Yes, that is as far as I'm going to go. So let's go ahead and look at what's next. We have the Simmons citrate. So the Simmons citrate, or the Simmons test, it looks for specific enzymes, and those enzymes are right here, the citrate amylase and the citrate lyase. Or you can be lazy like me and just say citrase. So like citrase. So citrase, that's completely fine. And I can't spell, as y'all know. Can't spell for the life of me. So citrase is also another way to put it. But those are the two enzymes that it looks for. And then the pH indicator for Simmons citrate is it's like bromethanol blue or something. It's like bro blue. Sorry guys, bad joke. So let me go ahead and pull it up so you can see what crazy word I'm talking about, since I can't speak English. Okay, so oh, I don't have it written down here. Okay, I will write it for you. So the pH indicator for Simmons citrate is I believe you spell it like this, bromethyl. Oh crap! And of course I wrote off the page like a dinkus. Okay, let me write smaller. I believe that's how you spell it. That's the pH indicator, and that's for Simmons citrate. And then if you are reading this test, ideally you will see a color change. So if your agar turns like a royal blue, like very blue, like very, very vibrant blue, it is positive for citrate. So that means that it uses it as a final carbon acceptor. And then that means that it's positive for it, so it uses it. But then if your if your slant doesn't change that royal blue, it kind of stays like a green color, then it's negative for citrate, so it does not use citrate as a final carbon source. So that's for Simmons citrate. Let's see what's the next one. So the next one is the urea. So urea uses, it looks for the enzyme urease, and the pH indicator is, I believe it's, what is it? Phenol red, if I remember correctly. So urea. So urea, like I said, it's looking for the enzyme. It's looking for the enzyme urease, and then the pH indicator is phenol red. And then what you're looking for is a color change. So if you have a positive urea test, it will be a vibrant pink, like I mean like neon pink, and that is positive. And then if there is no color change, if it stays kind of like a yellowy orange, maybe even some red tones to it, it is negative for urease activity, meaning that there is no enzyme present. And we saw some bacteria like that, and the bacteria that we saw like that were generally positive for urea was Klebsiella, and then we also saw P. vulgaris, and then also P. aeruginosa were the positives that we saw. So just keep that in mind. And then I believe next we have mannitol, right? Yep, we have malonate broth. So malonate broth, you're also looking for a color change. The pH indicator for that is the same as the Simmons citrate. It's the bromethyl blue for the mannitol broth. Mannitol itself is an enzyme inhibitor of specifically of the succinylate dehydrogenase. So it shuts down the Krebs cycle within a bacteria, and then also sometimes they can actually use that enzyme as a carbon source. It's kind of cool. But with malonate, like I said, you're looking for a color change. And if there is a green color to your broth, then that means that it's an acidic environment. And then if it changes to blue, that means it's an alkaline environment. And if it changes to blue, it is positive for the presence of malonate. So meaning that they are using malonate in order to function. So for malonate broth, you're looking for a color change. It uses the enzyme, the succinylate dehydrogenase, and then the pH indicator is the bromethyl blue. Okie dokie. So I believe that will wrap up all of our media types. Also remember, keep in mind positive ones. Also, I have all the pH indicators listed right here for them, just for y'all to know. So EMB uses the following, and so on and so forth. Okay, I already went over the zone of inhibition problem, so I'm not going to go over one, but I am going to write down that this ring that you see right here, sorry, that's like a terrible circle, I am going to write that is a zone of inhibition, definitely know that. And then also be able to interpret data that I give you from that based on this table, kind of like what we did in lab. So I'm not going to do that. Also be able to measure them. So like if I were to put a ruler on top of the zone of inhibition, I would want you to be able to tell me the measurement of zone of inhibition. So meaning that you need to know whether or not if it's in centimeters or millimeters, whatever it may be. So definitely know that. So be able to measure them. I will give you an image that already has a ruler on it, just so it's quote unquote to scale. So don't worry about that. Okay. And then based on the measurements, I would want you to be able to tell me the appropriate treatment plan, which is what we went over in lab the other day. So already good for that. And then, like I said, if I were to give you a flowchart or a dichotomous key, I would want you to be able to interpret that. So I'm actually going to go ahead and pull one up so you know what I'm talking about. So I'm just going to pull up. Okay, so if I were to give you this dichotomous key or flowchart on your exam, I would want you to be able to interpret it for me. So if you had, let's say that you ran a urease test, so a urea test, and based off of that, you had negative results. So I gave you, what I'll give you is that after you have interpreted all of the cultures that are like on your exam, you will have to interpret them and then use the flowchart. So let's say from your information, you had a urea test that was negative. So from that, you will go down here, and then from there, what will happen is that you will look to see if you had motility. So if you are looking at motility, you are looking at a sim test. So from a sim test, you would tell me whether or not if it was positive or negative. Now, if you have H2S production, you cannot tell. I'm going to tell you right now, you can't tell if you have motility. It's hard to see. So let's say that it was positive for motility, so that means that you will go in this direction because this is the positive route. And then from that, let's say that you had H2S production. You had a lot of H2S production. And then from that, you would be like, okay, so the species that that from all your results is a Salmonella species. So not too bad, right, guys? So let's actually try this again. So let's do it one more time. Let's say that we ran an indole test. And so indole means that you will have that red ring after you add a Covex reagent to your sim tube. So if you have a red ring, if you have that red color, then it will be indole positive. So that means that we're going to go here. And then from that, we also looked at a sim test that was H2S, let's say H2S negative. So that means that there was no black production in any tubes. So H2S production isn't always in sim tubes. Remember, it is also in TSIs. So if you have blackening in a TSI, that is also telling you that you have H2S production. But let's say we didn't have H2S production, so no blackening in any of our tubes. So that narrows it down to these two. And then from that, we did not do this test, but let's say that we did, and let's say it was positive. And if it was positive, then you get this bacteria. So that is what I would want you to do. Like, I would give you these cultures. I would want you to interpret them, kind of like what you've been doing with your unknowns. And then from that, use the flowchart that is provided on your exam and tell me what species or what bacteria do you have according to your interpretations. So that is pretty much a good chunk of the exam, guys. I believe that's it. Also at the bottom of your review, I do have some like big picture stuff that you can get from each lab. So remember, we're only doing some of them. We're doing the gram positive and the gram negative lab. We did a concept from the disinfectant lab, and then we're doing the concept of unknowns in terms of interpreting results and how to identify from a dichotomous key. So that is going to be your exam. If you have any questions, definitely contact. And in the meantime, happy studying, and I will see y'all in class.